Всім доброго вечора. Отож, за кілька хвилин відбудеться початок нашого ефіру. Залишається 3 хвилини, ще чекаємо трошечки і будемо розпочинати. Ефір буде англійською мовою, щоб це ні для кого не стало раптом сюрпризом. Dear viewers, in a few minutes we are going to start our lecture. Waiting 3 minutes. 3 minutes left. Good evening.
Доброго вечора, шановні колеги.
Доброго вечора. Good evening. Please tell somebody if you can see the presentation.
Yes, we can see it.
Thank you so much. I hope everything will be going on fine. And if some problems will occur, please tell me. Okay, so good evening, everybody, ladies and gentlemen, and welcome to the BIMCOvision. Greetings from Chernivtsi, Ukraine. So BIMCO 2021 is delighted to meet its past and future participants, partners, and friends online. And for those who have never taken part in BIMCO, we want to highlight the fact that apart from an academic activity, BIMCO is a vibrant community that values what each and every one of us considers. BIMCO, our scientific forum, is essentially a mosaic of unique visions on science, humanity, and art of medicine. My name is Olena, and I'll be the main host of this interview. And without the further delay, I'm excited to welcome our very special guest for today, Florencia Caro. And let me tell you something about our guest. Florencia Caro is originally from Argentina. She studied biology at University of Buenos Aires and then moved to the United States to do her PhD in the Derisi Laboratory at the University of California, San Francisco. After completing her PhD in malaria, Ms. Caro moved to Boston to start her postdoc in the Malacanthus Laboratory at Harvard on cholera. Florencia Caro is currently applying to academic positions, and her hope is to one day have her own laboratory. Thank you so much, Ms. Caro, for accepting our invitation.
Thank you so much for having me, Olena.
It's my pleasure. And our topic for today is acute and important. Malaria is a serious health problem. It is a leading cause of death in large parts of Africa, Asia, South America, Central America, and the Middle East. So, dear viewers, we invite you to join our discussion. Please write us your questions. Don't hesitate. Ms. Kara will be glad to clarify all the points you are interested in. So write the questions in chat. And at first, let's talk about malaria in general. What causes this this dangerous disease? What is epidemiology, pathophysiology of malaria, prognosis, etc.?
Well, first of all, thank you so much for the kind introduction. I'm really, really excited to talk to you about malaria today. I also want to encourage the audience to ask anything. I really, really love talking about this, and I think it's an important problem. So malaria actually is caused by a protozoan parasite called Plasmodium. Plasmodium falciparum, particularly the species, is the one that causes the most severe form of the disease. Plasmodium vivax is the second species that is also endemic to several areas of the world, but is less severe than Plasmodium falciparum. So I'll be mainly talking about Plasmodium falciparum today. So just to give you an idea, there are currently 200 million cases of malaria, active cases of malaria. And death by Plasmodium falciparum occurs mostly in children under the age of 5. And just to give you also an idea of how bad this problem is, every minute a child dies of malaria in Africa. And so for comparison, COVID right now, right, is about this is about a third of COVID deaths currently worldwide. But malaria has been causing this amount, this number of deaths for forever. So this is really a problem that is extremely relevant and that we have to address. But obviously, because it occurs in the developing world and places where, you know, the medicine and just in general treatment and therapy and vaccines don't reach, that's why it's still a very big problem. And I talk about children because so the mortality rate is highest. About 80% of the cases of malaria are children death cases, and the reason is because if you actually have an episode of malaria and you survive, you probably become rather immune. So there's some humoral response and also native immune response that prevents you from get reinfection. And you can, in places like Uganda, for example, you can get, I should say that malaria also is transmitted by the Anopheles mosquito. An interesting thing, it's only female mosquitoes that bite and at night and are infectious. And so if you get insecticide treated bed nets for sleeping at night, you probably can actually prevent this spread of disease. Yeah, and so I should probably also introduce the life cycle of the parasite. So the way it works is an infected mosquito injects sporozoites, about 10 to 15 sporozoites, into the skin. And then these sporozoites go to the liver, where they undergo several rounds of replication, and then they burst from the hepatocytes, this merozoite form that goes into the bloodstream. And then the merozoite infects a red blood cell, and the parasite then establishes its whole life cycle within red blood cells. It doesn't go back to the liver. And it's actually very interesting because it's a very synchronous kind of disease. So the parasite, upon invasion of the red blood cell, replicates and then bursts it open in a very synchronous manner. And then during this burst and lysis of the red blood cell is where the peaks of fever occur that are very characteristic and that happen every 48 hours. And so the amount of parasites that a person can have in their blood system at a time is up to 10 to the 12 parasites, which is extremely high. And then this is an asexual, the parasite at this stage is haploid, and so it's an asexual reproductive cycle, but then the parasite differentiates into gametocytes that are either male or female, and those are the forms that are taken up by the mosquito, and then inside the mosquito, the sexual cycle occurs. And so then the infectious mosquito goes on and infects another human.
So if it is end of the explanation, I suppose we can move to the most serious questions for today.
Yeah, definitely.
Okay, so what is the situation with malaria in the world right now? Maybe the mortality has increased or decreased or kind of that, and how the epidemiology has changed, and what is the situation right now at this moment with malaria?
So I think one of the more recent developments is the release of this vaccine that is actually only 58% effective, and GlaxoSmithKline has introduced it. The vaccine is made with an antigen that's present in the sporozoid form, the one that is injected by the mosquito. And so even though it's only 50% effective, there's been a very large push to put this vaccine out there and distribute it more recently. The Bill and Melinda Gates Foundation puts a lot of money towards these kinds of prevention. But, malaria is a preventable disease. If you don't get bitten by the mosquito, you probably won't get it. But in endemic areas, as I said, there's about 1,500 infectious mosquito bites that one person can get a year. So that's a lot. And so a vaccine would allow to, particularly for kids, to raise immunity against these infectious, the first infectious form. The reason it's so hard to create a malaria vaccine is because of its life cycle. And so the vaccine that I was just talking about is is against the first stages, but then the parasite, once it moves on from this stage, it never goes back. So the antibody response that you create is no longer valid. And so for the asexual reproduction cycle inside the red blood cells, vaccines against those kinds of antigens are being developed right now as well. But if you think about the sheer number of parasites you have to raise an immune response against with antibodies, it's a larger problem. And so that's one of the things. The other things that are a novel approach that more recently has been applied is this drug called artemisinin. That's a drug derived from a Chinese plant and has been shown to be very effective against the asexual stages of malaria. So it's used usually in combination with other drugs that are like chloroquine and other types of drugs. And the reason it's used in combination with these drugs is because there's a lot of resistance. These drugs have been used, chloroquine, for example, has been used for years and years. And so the parasite has adapted and become resistant to them. And artemisinin, because it's a new drug, there's not much resistance yet. And so the problem with artemisinin and why it's not so great is that the half-life inside the body upon ingestion is only 45 minutes. So you really have to use it in combination with other drugs in order to really curb the disease. What else were you asking? Yeah, I guess if you have any questions.
Dear viewers, yes, we have some questions in chat. Florencia, if you can, or I can read them.
Yeah, I actually saw the question. It's a very good question. Absolutely. Because the vector is the Anopheles mosquito, you really have to have the vector present in order for transmission of malaria to occur. And in the northern hemisphere or further south, the temperatures for the mosquito are such that they're not viable during the winters, for example. However, there has been occurrence of malaria in the developed world, but because of the availability of resources like insecticides and just vector control in general, the malaria in the developing world is not a problem. The hygiene, I don't think it's necessarily poor hygiene, but because mosquitoes' breeding grounds occur in marshes, right? Like water that's been stagnant, that's where mosquitoes like to breathe. In general, you associate, places with low infrastructure with higher amounts of mosquito. Another thing that's interesting is that malaria can occur, and usually frequently occurs in these endemic areas, in conjunction with other diseases. It's very, very frequent that you will have HIV positive and malaria cases, or even multiple species of malaria infecting one host. So it's a combination of problems that occur in the developing world that make it so hard to curb this disease.
Okay, I'm so sorry. We have another question from Ms. Sidorchuk M. From Professor. Can you hear us? Anyuta Stepanivna?
Good evening. Yes, thank you so much for opportunity to be present here. And I have a brief question about your vision, Florence, according to malaria treatment approach for pregnant lady. What can you say? What is your statement? What is your idea? Thank you.
You're asking in relationship to pregnancies, right?
Yeah, right.
Pregnancy, yeah. Interestingly, the parasite can cause what's called placental malaria, right? So the Plasmodium falciparum has the ability to both cross the blood-brain barrier and go into the placenta. So yes, placental malaria is a very big problem. I don't know much actually about the current treatment and what the WHO has in terms of protocols established for pregnant malaria patients. But I do know that pregnant malaria, placental malaria is associated with miscarriages. And so, but yeah, again, I am not very much trained in the clinical aspects of malaria. I'm more of the molecular biologist. I'm sorry if I couldn't give you more information about this.
Thank you so much, Florence, for answering questions. Okay, so may I can move?
Yes, sorry, are you asking me if you can move on?
Yes, okay, I suppose I can move. So the next question is, which factors determine the virulence and severity of malaria?
Yeah, so the reason one gets so sick is because, as I said, every 48 hours, the majority of your red blood cells are bursting. This means a severe anemia ensues. And so then the parasite is released, and that causes also a lot of, there's endotoxins and just high peaks of fever. But also what's fascinating about the parasite is once it invades the red blood cell, it actually specializes in expressing certain parasite proteins on the surface of the red blood cell. And so these parasite proteins do tend to have some cytoadherent properties. And so what happens is upon traveling to the bloodstream, parasite-infected red blood cells will actually adhere to the endothelium, causing clogs. And this is so that the parasite does this so as to prevent clearance through the spleen, right? So red blood cells are usually cleared through the spleen, but the parasite adheres and clumps at capillaries in order to prevent passage through the spleen. And so these clogs obviously can cause organ failure and that's usually also a cause of death. I really like the parasite is extremely smart, because obviously these are antigens that the parasite is presenting to the immune system, but the parasite is smart. So these antigens are called antigenic variation antigens, and they're encoded by 60 genes in the genome of the parasite. And these 60 genes are all similar, but have a little bit of a variant. And so every time a parasite infects a new red blood cell, it will express a different variant of these 60 different types. And so the immune system is continuously mounting a response to the first one, but then upon a second cycle, it's no longer useful to express antibodies against this antigen because the parasite has switched. So it's antigenic variation, antigenic switch. So the parasite is extremely smart at evading the immune system. Additionally, it is hiding within the red blood cell, right? And so the red blood cell, if the immune system attacks the red blood cell, there's also negative consequences on the body. Yeah, so I would say that, and then, yeah. I think those anemia, severe anemia, capillary, and then obviously the parasite crosses the blood-brain barrier, there's cerebral malaria, and that usually is fatal.
Such an amazing thing that such a small organism can be so clever and
yeah, and it's a fascinating also organism, because if you think about it, so what's interesting about these apicomplexans, right? This genus is also like Toxoplasma is a similar parasite, is that they have inside of them, what differentiates them from other parasites is a plastid, and this plastid is originally used to be a cyanobacteria. So it used to be, there's vestiges of photosynthetic genes. So these parasites are very, very weird in that regard, in the genetic composition, because they're eukaryotes with some photosynthetic ability. And they have, aside from this plastid, they have these apical organelles that are at the top of the parasite. And once a merozoite finds a red blood cell, it kind of rolls over on the membrane of the red blood cell and then orients the apical organelles towards the red blood cell and releases a lot of proteases and things in order to open a space for the parasite to go in. And then inside the red blood cell, the parasite lives inside a vacuole. So there are multiple membranes that separate the outside from the parasite. What you're seeing in this image actually is these are Giemsa stained red blood cells, and the two dots that appear on the right of that would be an early stage parasite. You're seeing the genome, the DNA of the parasite stained with the stain. And the two other darker things that you see are actually gametocytes. These are the sexual forms of the parasite inside the bloodstream that are the ones that the mosquito will take up. Yeah, it's a very, the other thing that's very interesting to me, at least, is the parasite is oxotrophic for the majority of the amino acids. This means that the parasite cannot synthesize amino acids itself. It has to take them up from the host. And obviously inside the red blood cell is hemoglobin. And this is a protein that has every amino acid except isoleucine. So the parasite has to take that from somewhere else. But the other amino acids, it can just degrade hemoglobin and then eat it up and then grow. However, within hemoglobin, there is this prosthetic group called heme, right? But heme, upon degradation of hemoglobin, is actually toxic to the parasite. So the parasite has devised this ability to polymerize heme into what's called a crystal, a hemozoin crystal. So these are polymers of heme. And when you look at them under the microscope in late-stage parasites, you can see these dark stone, like a little stone of hemozoin. And one of the tricks that we use in the laboratory in order to isolate malaria parasites, late-stage malaria parasites, is simply putting a magnet against the laboratory tube, and the parasites are paramagnetic due to this hemozoin crystal, and they will bind to the magnet. And so that's a way for us to purify away a parasite in a tube of a complex mixture.
Amazingly.
Yes, it's very exciting. Yeah, it's very, very cool.
Excited. So we missed one question. We know that you are learning expression of Plasmodium genes, and can you explain what does expression of genes mean for maybe somebody don't understand, and what methods are using to investigate gene expression, and is it possible to affect gene expression, and how?
Yeah, this is actually what I am trained in, and so this is more my domain. So gene expression, you all know there's DNA, right? And then within this DNA, there are genes, and these genes got turned on to express an mRNA and messenger RNA, and this messenger RNA will then get translated from the ribosome into a protein. And so basically gene expression is, depending on the stage of the parasite, you will have a certain amount of genes expressed that will encode proteins with functions that are relevant to the parasite at any given stage. And one of the things that are extremely interesting about the malaria parasite is that if you look at it has about 5,500 genes. So that's 5,500 genes encoded in a genome that's 14 chromosomes and I think 32 megabases. So these 5,500 genes, each of them encodes for functions that are relevant to the parasite. And I'm only talking about the sexual stages, by the way, because that's what I studied. And I would say that those stages are very relevant because that's where the disease symptoms of the disease occur, right? And so once the parasite enters the red blood cell, it begins transcribing a subset of genes that are relevant to the parasite in this early stage. Then those genes get turned off, and another set of genes get turned on, and so on. And each gene is expressed only once during the life cycle. This is very different than, for example, how bacteria work, right? Bacteria have also, say, for example, cholera encodes 3,600 genes. And they are on and off depending on what the bacteria senses in the environment. But malaria is very set. In fact, this genetic program of turning on a gene once per cycle is so fixed that in the laboratory, you can increase temperature, you can perturb the environment of the parasite in a tube, and the program will not change. So it's a very rigid transcriptional program of turning genes on only when they are needed. It's very different than other species or unicellular organisms out there. And so once a gene is expressed and the function of the gene is translated into a protein, then what I did during my PhD is ask, well, perhaps the reason we're not seeing that the parasite responds to environmental stimuli is because every control of gene expression happens after transcription. So you turn the gene on, but perhaps after that, there's ways of regulating how much of the gene gets translated. So the way we asked this question was by measuring translation on a genome-wide level with this new technique called ribosome profiling. So the ribosome, right, is a megadalton complex that sits on a messenger RNA and translates the gene. And so you can arrest ribosomes on a messenger RNA and then purify this polysome. When multiple ribosomes are sitting on a messenger RNA, it's called a polysome. You can purify this polysome and then cut right where the ribosome was sitting, and this will create a footprint of where the ribosome was sitting at a time. And then you sequence using next generation sequencing technologies. And Elena, let me know if you want me to explain how next generation sequencing works, but perhaps people are familiar with that. You basically sequence everything that you get that was sitting under the ribosome. And so you have an idea, a snapshot or a photo of exactly what is being translated at the moment you harvested these polysomes. And so that gives us an idea of whether there was or not regulation of gene expression at the level of translation. And I can tell you a little bit more about that data. But I think what's interesting is that when I was doing my PhD, genetically manipulating malaria parasites was very hard. So the way you actually work with malaria in the lab is I used to donate my own blood to put the parasites in. And so you have your own blood, you have a flask, you put nutritious media on top. And so it's blood plus nutrients. And then you can propagate this, giving the parasites always new blood in order to reinvade. And at the time, we didn't have many good techniques to genetically manipulate the parasites, so delete genes, edit genes, in order to study it, because it's a very finicky, as I said, there are multiple membranes that you need to transpass in order to reach the parasite. So genetic manipulation is hard. But because of CRISPR technology, are you familiar with CRISPR technology, Elena? There's this new technology that's called CRISPR. Now everybody can manipulate parasites. So it's been a revolution in the recent years because people have been able to actually interrogate the genes in a way that was previously not available to us. Yeah. So there's a lot of new methods currently being used, and I think that's very exciting.
Whoa, complicated information, I should say, and that's because I just have to ask our auditory. Maybe somebody has some questions. Because really my brain are shocked with this information.
That's a lot of stuff.
Okay, if somebody has questions. Okay, we have one question. Florencia, can you?
Yes, the question is, why do people with sickle cell anemia not suffer from malaria, and how does the sickle cell erythrocyte influence the penetration of Plasmodium? Yes, so the sickle cell anemia and also other blood, genetically, hereditary blood problems are actually positively selected in populations where malaria is endemic because, as the question said, the parasite has a harder time penetrating the red blood cell. The membrane of the red blood cell, for example, in thalassemia and sickle cell disease, is more rigid and it has different properties than a normal cell. And so the parasite cannot penetrate it very well, and these people are protected. However, obviously, homozygote, so having thalassemia and sickle cell disease is usually a heterozygote phenotype for these humans because it tends to be very people with homozygote genes are very sick. But the heterozygote phenotype or people are selected positively evolutionarily because they are more resistant to the parasite. Yeah.
And we have another question. Can you see it, Florence, or
yes.
Should I read?
people who are not native from African countries, but living for a long period in the epidemic malaria, they get immune protection against malaria. What do you think about this? Is it possible? Is it myth? No, absolutely. You become immune. As I said, people are exposed to malaria at least 1,500 times in a year in Uganda, for example. And so you do raise this. This means two things: that the immune response that a human can raise against this parasite is protective. And so this means that creating more vaccines is an avenue for prevention of this disease. So it's good. We have to just learn more about what kind of immunity you need to raise. And then the other question is, what kind of affordable anti-malaria protections while you're traveling? So that depends, I think, on the area that you're traveling in the world, because depending on which region in the world you go, there's different resistant strains of Plasmodium falciparum. So chloroquine, for example, resistant parasites have been spread all around the world. So people tend to not use chloroquine anymore, or if they do, it needs to be in combination with other drugs. And I do know, however, that these anti-malarial prevention, like chemopreventative therapies, tend to be very toxic. You know, they have side effects. So it's important to also weigh both things, not to take medicine if you don't need to take it. And additionally, if you are going to get a treatment for prevention of malaria, you should make sure that you follow it through, because otherwise, if you don't do the full course of antibiotics, you are also making the parasite be prone to acquiring resistance, right? So in terms of affordability of the antimalarials, that's a big issue, because the new artemisinin-based therapies tend to be very expensive, particularly for people in the developing world. I think a dose currently goes at $8 per dose. So this is not an affordable strategy for many places. So governments are subsidizing, and obviously nonprofit organizations like the Bill and Melinda Gates Foundation put a lot of effort to making these antimalarials available. Yeah. Is there any way to prevent the expansion of malaria to northern regions with climate change? Oh yeah, that's a very important question. Particularly, and there was a question I had, right? Climate change will make the regions of the world that are currently the mosquito will probably thrive in more areas of the world. But again, this is a preventable disease. If you have money, you probably will be able to curb it. Why does Plasmodium vivax persist in hepatocytes for a few years on the silent stage? Yeah. You know, I don't know how. So the question, so Plasmodium vivax is that other species that is less virulent, but actually can persist in the liver in this form called hypnozoid. So you have no symptoms of disease, but you are not clear of the parasite, because the parasite rests in this dormant stage, so it's metabolically inactive, and it stays there inside the liver for months at a time, and then sometimes on occasion it leaves the liver and causes disease. So yeah, it's very interesting. I don't know the molecular or the mechanism behind this kind of dormancy and what makes it revive. But I do know that obviously this is bad, because the morbidity that this causes, to have this latent malaria always is bad for humans. I don't know, right. So how would you clear a Plasmodium that's in the liver in a dormant stage? It's a really hard problem to tackle, because in a dormant stage, any cell is not prone to picking up any of the drugs or metabolizing the drugs in a way that is harmful to the parasite. So I don't think there's strategies to killing parasites in the liver that are dormant currently. what role does the spleen play during malaria infection? How does this contribute to some of the hallmark symptoms of malaria? So, as I said, the spleen clears red blood cells, but the malaria parasite cheats the spleen, so cheats the system to sequester red blood cells that are infected away from the spleen. So, red blood cells infected with malaria generally don't tend to go through the spleen, otherwise the parasites would be killed in that stage. So they prevent that by adhering, using these adhesion molecules to the endothelium of the capillaries, so the vasculature.
Okay, so I suppose we can move. Okay, this question, I suppose, was answered about prevention, isolation, and I suppose we can move also. And the next question is, how does this knowledge can be used in understanding of pathophysiology of malaria and in treatment?
So I can tell you what our findings of my graduate work. So what we found was that the parasite applies regulation at the level of translation in the very late stages of development inside the red blood cell. So one parasite infects a red blood cell, grows, and then divides and makes about 30 to 36 copies of itself. And these are 36 merozoites, and once they are ready, they burst open the red blood cell, and then they go. And what we found is that these merozoite stages have transcribed genes, but have not yet translated them into proteins. And these genes are all necessary for the stage that comes as soon as they find the red blood cell. And so it's a very neat strategy to be ready. The merozoite will be swimming in the bloodstream until it finds a red blood cell. And as soon as it does, it will initiate the translation of these genes. So it's ready to go. And that's a faster strategy for gene expression than having to transcribe into mRNA first. So the regulation at the level of translation in these stages appears to be the important part. So we are, obviously this is basic research, and so the direct application of our research into the actual prevention of malaria, there's always a big step, but basic research is important, I think, to precisely understand how this parasite works. If you think about it, there's only 5,500 genes, but for 60% of the genes in the malaria genome, we have no idea what they do. So unknown functions, they are annotated, and we don't know what they do. And I think a deeper understanding of the functions that this malaria encodes is important in order to figure out novel strategies to prevent and drug this parasite. One thing that I forgot to mention that I think is fascinating too is that most of the DNA in the majority of living species is about 50% AT, 50% GC, so AT, GC at equal representation. The malaria parasite is 80% AT. That means that if you look at the genome, you will see AT, very, very low complexity, AT-rich genomes. And this is actually, for molecular biologists like me, a puzzle, but also harder to work with, because usually genes either begin with ATG and stop with a stop codon. But if you have such rich such AT richness, it is hard to figure out where a gene starts and where it stops. And so that has been also very puzzling for investigators throughout years. Why does this parasite have such a high percentage AT? Yeah, just a little snippet of information.
Okay, that's a large blast of information. I suppose I should in the next day look at all lecture again, because I'm just shocked with this information.
Maybe
Such a detailed, really. I need a book from a book of yours in the topic of malaria. Okay, in order to read all these details and understood them better. And maybe somebody has some questions. Don't hesitate, you can ask. Just let's wait a few seconds. Maybe somebody is typing. And I have a question. Maybe auditory would be interested in your work. What are you doing in laboratory? What news are in laboratories? Maybe something that we will read in books later. Some interesting pieces of your work.
Yeah, as I said, I'm a molecular biologist by training, so I like to manipulate genes and make genetic changes, and currently I'm working with the Malaria, and so we apply just different methods to perturb. One of the biggest questions, I think, with every pathogen that scientists try to answer is, what are the genes included in the genome that are absolutely essential for survival of this cell? And it's not all 5,500 genes in malaria are necessary for its survival. You can actually probably delete half of them, and the parasite, make individual deletions, and the parasite will be fine. So it's always interesting to us to figure out what is the component, the essential set of genes that without their function, the parasite or the bacteria will die. Because those are the functions that we need to know in order to make drugs to target those functions. Because if you make a drug to target a non-essential gene function, that is not going to kill the parasite or the bacteria. So I've always, throughout my career, I've been looking at what is the genetic background, the core genome that an individual cell of a pathogen requires for survival. And then the next question that I always have, because it's just interesting to me, is why are these essential, right? Why would you need this particular function encoded for survival? And so some are obvious, DNA polymerase to replicate your genome, but then there's others that are not so obvious, and that's where I chase those answers.
Amazingly, I have never even thought about that. It's just a new opening for me, and I'm so sorry if I look all the time downwards, because in my computer screen there's only presentation, so that in order to see you, Florence, I should look in my phone. My phone screen. So sorry.
I don't mind.
Okay, dear auditory, if you have some questions, it's your time to ask. Please.
Could CRISPR be used to create an immunity to malaria other than sickle cell anemia? And that is a very interesting question. Can CRISPR used to make humans resistant to malaria? And I would say that the answer is probably no, because, for example, COVID, right? We know that there is one receptor for the virus to enter the cells, and that's ACE2. I think ACE2 is a receptor on many cells in the epithelium of humans, and perhaps with CRISPR technology, you could kill that receptor. I don't know if it's if it creates other types of problems, but then you would eliminate the entry of COVID into cells. For malaria, it's more complex than that because you don't have one receptor for entry. Additionally, malaria has multiple types of stages and forms that it embodies, right? It goes through morphological changes. COVID is just one viral particle. It's much simpler. So I don't think CRISPR, like genetic manipulation of humans in order to prevent malaria infection, is feasible, but a very interesting mental exercise. And then, but just to say one more thing about CRISPR, I think it's the future of any. We should focus on this technology because it definitely is the answer. Is our molecular tool to generate more tools for us to combat very, very important diseases. So yeah, CRISPR is important. What imprint does Plasmodium have on a human body after recovery, and how can you understand that person has already had malaria? That's a very interesting question, actually. I think, well, certainly you will see antibodies that you can find, particularly to these antigenic variation genes that I mentioned earlier. So PFEMP1 is that one protein that the body responds to, so antibodies against malaria. And the sequela on people with malaria, I think that once you clear the parasite, I don't know that you have any problems afterwards. It really doesn't seem to be a big problem for adults that have already a very strong immune system. It really just causes severe effects on children under the age of 5. So I don't know that there's morbidity associated. But obviously, if you get infected with malaria frequently and you don't have the proper immunity, it's just like you're constantly sick. That impacts your ability to lead a successful and healthy life. And so I think that's it. Yeah.
So another question, maybe. Guys, don't be shy, even if your question seems for you to kind of obvious, maybe stupid, or I don't know what can stop you. But you are welcome to ask. You are here to know more, and we are here to tell you something new.
Yeah. I'm happy to talk about anything. This is one of my favorite topics.
Okay, so let me ask you a question. Why have you chosen malaria to investigate more detail?
Yeah, I always wanted to study biology. I'm fascinated with genes and genome and how everything that, like every living thing, has basically one molecule that we all share, from plants to bacteria to humans. It's just DNA, and within that DNA, information to building an organism is encoded. And I think that's fascinating. And so then I just wanted to learn more about genes when I was little. And I also wanted to think, well, how can I apply all this knowledge to something that is relevant for the world? And I think neglected tropical diseases is important because obviously cancer and other diseases that affect the first world there's so much money put towards research on these things, and already there's cancer therapies and things like that are continuously being developed. But I don't think that tropical diseases that affect people in the poorest regions of the world are given the attention that they merit. And so I just wanted to both have fun with what I do at work every day in the lab with all my tubes, and think about that perhaps my research will have one day some sort of positive impact on people that are less lucky.
What a noble thoughts. Really, I'm amazed.
I don't know, because again, I'm far removed. I think people like you, Elena, who study medicine and are directly involved in curing people, are also, it's a noble pursuit as well. For me, I'm very far removed, but I like to think that my everyday activity will one day be relevant for people in the world.
I'm sure that your everyday researches will be relevant. I'm sure that my kids will be reading about you in their books. And
I got a question. Florida mosquitoes, 750 genetically manipulated mosquitoes. I love that too. Yeah. How dangerous can it be, and can they spread to some GM mosquitoes in such a way? So I really like that, because one way of curbing the spread of malaria is obviously addressing the vector control, right? Oh, my son just woke up from a nap. Milan, I'm at work. You have to go with daddy, please. Sorry.
Oh,
honey, it's okay. I'll be right there. I have to finish talking to my friends. Okay, I'll be right there. He's 3 years old, woke up from a nap. Okay, so the way this technology works is you make the male mosquitoes, I think, sterile, and so they cannot propagate. Again, people are very, very worried about spreading genetically modified organisms in general, but I believe that it's an excellent strategy, because as the next question asks, for example, vector-borne, mosquito-borne diseases are like dengue fever, yellow fever, all these other ones could be also cured by the same exact strategy. Get rid of mosquitoes, you get rid of diseases. I wouldn't be so worried about the consequences, to be honest. Obviously, we cannot know what the consequences are on ecosystems that depend on mosquitoes, for example, other organisms that eat mosquitoes as food. But I think what's the problem with creating a perfect vaccine? And could you have cross-reactive vaccines? Yeah. Again, the problem with the vaccine is that the parasite has many stages, and you do have to mount a very, very strong immune response, because once the parasite is in the red blood cell, it really makes replicates of itself so much. But there's a lot of vaccine candidates currently in the pipeline, but only this one has just made it to the market, and it's only 5%, 58% effective, and only in children above 12 months. So the most vulnerable newborns are not protected by this vaccine. So I don't really know exactly what the problems are with generating an efficacious vaccine, but there are a lot of them in the pipeline. In accordance with WHO, a newly created vaccine was already applied for malaria prevention in African regions. Have you heard about its official? Yes, that's the one that I was talking about. It's called, it's a GlaxoSmithKline vaccine against RTSS, which is that antigen that sporocytes display on the surface.
I also have another question. Why malaria has the worst impact on children? Because I saw the facts in presentation. Maybe I missed this information when you was talking about that kids suffers from malaria the most.
Yeah. Well, that's it. It's because kids have not yet developed a strong immune system, right? And so they're more vulnerable, and also I think there's other problems that are happening at the same time in these regions of Africa. Perhaps the child is malnourished or doesn't get the attention immediately. Sometimes the child may, like early intervention of the disease before, once the symptoms occur, if you give the drug therapy right away. But what happens is many people don't even have access to fast, drug treatments. And so it really exacerbates the problems. We don't know if you had, so there's a lot of effort in trying to deploy trained physicians to local endemic areas that could provide fast access to drugs, and immediately also be able to identify what kind of species of malaria it is. etc. But training, access, it's just a combination of a lot of problems that causes this high mortality in children, really. And artemisinin are the most efficient ones, but one of the problems, as I mentioned earlier, is that the half-life inside the bloodstream is only 45 minutes, so you really need to put other drugs in there, and the parasite becomes resistant. So that's one basically of the problems that we're currently
How
facing.
terribly poor kids.
Yes. We need to develop more drugs, different drugs.
And another question in chat: how efficient is
Yeah, that's the one.
artemisinin combined therapy in combating malaria?
One of the things, so everybody really likes the ACTs, they're called artemisinin combination therapies. But one of the recent things that people have been seeing is that in Southeast Asia, there's this phenotype that's been appearing in people that take artemisinins, where it used to be very efficient at clearing the disease, and now it's not as efficient. So it's not that the parasite is resistant, but the parasite takes longer to clear. And that is an alarming problem. And people don't want this kind of resistance to spread to the rest of the world, because really artemisinins are the only frontline drug treatment that we have. And so the mechanism of artemisinin resistance is not very well understood. Chloroquine resistance, on the other hand, is very well understood, because the parasite expresses this efflux pump. So chloroquine goes into the food vacuole of the parasite, but the parasite flushes it out, takes it and flushes it out. So that's the resistance. But artemisinin, we don't know exactly the mechanism of resistance, so we don't know what to look for. It's just this one delayed death phenotype that we're seeing, and it's very, very concerning, because if this resistance spreads around the world from Southeast Asia, which it probably will, if not controlled, then we are back to square one. No real good drugs. So that's the current status with artemisinin.
Okay, guys, maybe some other questions. We're having a warm chat, I suppose. So you are free to ask. Okay, so if there is no more questions, that brings us to the end of questions hour for today, and I should say, Ms. Carol, thank you so much for being here with us today and giving us the pleasure of hosting you. The information you shared with us today is really, really deep and has lots of points to think about. And okay, another questions. I should stop my speech. And what is the best approach in diagnosis of congenital malaria in non-epidemic areas?
Is your question, let's say somebody gets to the hospital, what's the first way of detecting that you have malaria? Elena, do you think that that's a question? They want to know how to diagnose malaria if you're in a non-endemic place. If that's a question, I think that the symptoms of malaria are extremely clear. It's a deep anemia, very severe anemia, fevers that are extremely periodic. That's why it's important to take a fever periodically for the patient. And then the very easy diagnosis is you take a little bit of a blood, you make a smear on a slide, and you stain it, and you look, and you will see parasites for sure. A very well-trained microscopist like me will be able to detect the different stages and additionally identify which species of Plasmodium it is, whether Vivax or Falciparum. But sometimes, like, you have to have a lot of training because it's visually not as clear sometimes. But it's very easy to diagnose malaria. It's one of the, I would say, that should not be the limiting factor.
Thank you so much. I suppose we can end our past, and we can end of our questions for today, and let you to have your weekends.
Thank you.
And thank you, Florence, so much for
Yeah, this
coming.
was great. Thank you so much. I really had fun. I don't know if I was able to answer all your questions, but yeah, I thank you so much for the invitation.
We are really glad to meet you personally. I'm just really happy to meet you, to know you, and your knowledge is really just amazing, because I really you lighted up the points I have never thought about, and really will mind it in my future investigations.
Well,
And
I'm glad I could provide some of it.
Really, thank you so much, and thank you guys for coming, and for watching us, and see you. I suppose I should turn the slides. Thanks everyone for being with us today, and stay tuned for our next live session. Details will be announced soon in our Instagram page, BIMCO 2021. Follow us and know all the news the first.
Thank you.
Okay, thank you so much, Florence. Have a nice day. Have a
Thank
nice
you.
Thanksgiving weekends.
Yes. I appreciate it very much. Thank you.
Thank you. Bye bye.
Bye bye.